Chapter 4.5 is about what we call the cytoskeleton. So cyto means cell, and skeleton, you know what skeleton means. It is a skeleton, an internal structure. So this is the cellular skeleton, basically. There are different components of it, and different organisms have different systems. We're going to talk about three different types: microfilaments, intermediate filaments, and microtubules. We're going to compare and contrast some components that are involved here. Cilia versus flagella. These are involved in a couple of different processes, mainly movement. And then we'll talk about some differences in prokaryotic cells, animal, and plant cells here. So what is the cytoskeleton? Well, it's a network of protein fibers that help the cell maintain its shape, keep organelles in position, allow the cytoplasm itself and vesicles to move around within the cell, and in some cells, it enables the whole cell to move. The protein fibers that are in here are grouped based on size and composition. We're not going to get too deep into it, but I do want you to have heard of these things. Many of them are very important and ubiquitous throughout life, so they often get referenced. Microfilaments are our smallest cytoskeleton components. They're small, micro, and they're small filaments. The next up in terms of size are intermediate filaments. We'll talk about those. And then the larger ones, the largest of these ones are microtubules. And we will see what these can do in cells. So let's start at the bottom. You don't need to know the actual size, but you do need to know that these are the smallest. Microfilaments are the smallest cytoskeleton element. They are around 7 nanometers. They're made up of this protein called actin. They're a bunch of monomers that get stuck together to form these polymer filaments here. And you can see these different actin monomers coming together to build a filament here. They can participate in movement of the cell, in like cell division. They help the cell move and pull apart. And also in plants, there's this process called cytoplasmic streaming, where things can move around through the cytoplasm, and microfilaments play a role in that. In muscle cells, there's a lot of microfilaments. They actually move across each other to help muscle cells contract or retract in the process. So let's look at what that means. So here's an actin filament, and here's another actin filament, and then we have this what we call myosin filament. So you might know myo is muscle. On these, we have sites where these basically myosin molecules can attach little kind of motor proteins, I guess we could call them that, that kind of pull along. You can see here they're pushing and pulling along the actin filaments that can help muscle cells move. The signals that are involved in that, you can learn about those in anatomy and physiology. There's ions that are involved and electrical impulses, but the actual motor itself is this little molecular machinery down here that's made up of microfilaments. Okay, intermediate filaments are just that, intermediate in size, 8 to 10 nanometers. These are structural components that help keep cells shape and keep organelles in their place. They include fiber proteins like keratin. Intermediate filaments are technically not really found in plants. I taught a cell biology course one time, and I have to make a confession. I made the mistake of saying, I don't actually know a lot about intermediate filaments because I never had to deal with them. It's not true. I know things about them, but I was trying to make the point that plants don't have these. So like when I learned plant cell biology, we didn't even talk about these. That's interesting, right? Because animals evolved these, plants didn't. We tend to think their cells are very similar, but they have very different structures in here. You might know keratin as a protein that's involved in other structures like your nails and horns and hair and things like that. So it can form these filaments at a very small scale as well and be very strong in that process. Okay, if we go up to our biggest components, we're looking at microtubules. Microtubules are technically these big hollow tubes. I say big, they're 25 nanometers in diameter, so not big, but biggest of these. And they're made up of different monomers of what we call tubulin. Not very original name, but we have alpha and beta tubulin that winds round and around to form these tubes, and they kind of can act like a rail system. Vesicles move around on these microtubule rails, and I'll show you an animation of that in just a sec. They're integral in helping actually pull apart the chromosome pairs in cell replication, and they form some of our motile structures, things that can move cells around, like flagella and cilia, which we'll talk about in just a sec. So here you can see we have a stain here of our cytoskeleton components. Looks like the microtubules are stained red here, and we can see them moving throughout the cell to form kind of like a rail network. All right, let's take a look at these microtubules in action. All right, so here you can see an animation that I'll animate here in just a sec. We have the microtubule down here on the bottom, and here's some sort of vesicle, and there's this cool like motor protein here that is going to move this vesicle along. So here's an artist's animation of what is occurring in the cell. All right, so this is what it actually sounds like in the cell, of course. Not really. Okay, right. This is funny. But this is actually what the motor protein does. It basically walks along the microtubule and pulls the vesicle with it. So this is a very cool way that we can move vesicles around the cell. They don't just move by magic. They are moved by these motor proteins, and that does cost energy to move them along. But super cool little way to move stuff around. And you can actually watch this in real time if you have fluorescently tagged vesicles. You can watch them move around the cell. You can't see it quite on this detail, but we've inferred this is how it's working. All right, so that's one of the uses of microtubules, moving things around. Another one are these structures that are called flagella. Now, flagella is actually a non-specific term. You might think flagella, you might think sperm cells. And yes, sperm cells have flagella, but you also might think flagella, and you might think bacteria. Well, they serve the same purpose, but they actually evolved independently. In bacteria, flagella move in kind of a corkscrew-like circular pattern to, like a propeller to drive the bacteria through a liquid. In eukaryotes, they move in a whip-like fashion to move things through the liquid. These systems evolved independently, and I think flagella are one of the coolest looking things. You see this cartoon diagram here, right? And it kind of looks like a drawing of a motor or something that we might make, right? There's a rotor and a stator here, and it pumps hydrogen ions through and it spins this thing. If you look at it under an electron micrograph, you might think that this is a machine that we have built, right? But this is the actual structure of a flagella under an electron microscope. Super cool looking thing, right? And there are all kinds of cool uses of different microscopy techniques to figure out what is going on in here and build 3D models of this thing. So I want to show you this because biology is not just chemistry or going out in the field and counting birds and things like that. There's a whole field of physics and computation and math and computer science and animation and 3D modeling that can go into biology. So here's a 3D model of a protein, right? That's super cool. If you're into 3D modeling, you have valuable skills. And if you know biology, you can apply them to these systems. Whereas like me, I can play in Blender and do some 3D models, but like I can't make something like this and animate it. So those are valuable skills to have. So as I've always said, right, making connections between different things is really valuable. So don't ever forget that. Depending on your skill set, you might have special skills that people need. So flagella are one way of moving around. Another one are cilia. Cilia are smaller. So in the case of flagella, usually there's one, maybe two or three of those. Cilia are more numerous and shorter. They're little hair-like projections, and they all work to beat in unison to propel either the cell or to move things near or around the cell. So an example of cilia is here on this little single-celled eukaryote. These cilia will beat and it will swim through the water by pushing the water away from it. Here, we might have some cilia. This might be in the lung passage, in the airway. You have a thing called the mucociliary escalator, where all these little hairs beat to pull dirt and phlegm and stuff out of your lungs, and then you hawk it up and spit it out, right? So that system is cilia that are moving things along there. Table 4.1 in your book has a great overview of all these different structures that we talked about. You might take a look at that while you're studying. The cytoskeleton, at its core, is composed of 3 different elements. From smallest to largest, we have our microfilaments, intermediate filaments, and microtubules. We'll come back to some of these in just a moment, particularly intermediate filaments, some of their uses in holding cells together. Microfilaments, composed of actin, allow cells to move in places like cellular division. Intermediate filaments help keep the cell shape. They're fiber proteins like keratin. Then our microtubules, they can extend and retract to do things like help pull chromosomes apart or act like that rail system to move around vesicles. They form parts of the flagella and cilia to help cells move. All right, in 4.6, we're going to look at some connections between cells. In a multicellular organism, you have to have ways of holding your cells together, otherwise you just fall apart, right? So we'll look at that in the next section.